हेलो, हाँ जी सर। हेलो। नहीं, अभी तो मैं एमबीबीटी ले रही हूँ। हेलो। हाँ जी सर, मेरी तो अभी लेक्चर स्टार्ट है एमबीबीटी का। हेलो शिवानी मैम, गुड गुड इवनिंग। क्या हाल है मैम? एक्चुअली मैम परेशान होने की तो देखो, मैं अब मैम देखो, पहले मेरे 4 बजे से मैं बैठी हूँ कॉन्टिन्यूसली ना, पहले 4 से मेरी 6 थी, फिर 8 से अभी 9:15 तक थी, अभी फिर मेरी एमबीबीटी भी है। तो देखो मुझे भी जब वो अपनी साक्षी गई है तो मुझे भी एकदम से जेनेटिक्स मिला ना, मैं उसके लिए प्रिपेयर नहीं थी। तो उस चीज़ के लिए मैं पीपीटीज बना रही हूँ 2 बजे से बैठ के, 2 बजे से मैं ऊपर हूँ, सारे घर वाले नीचे हैं। तो मैं उस चीज़ के लिए बहुत तैयारी प्रिपेयर कर रही हूँ, क्योंकि मैं जैसे साक्षी छोड़ के चली गई एकदम से बीच में कि मैं तो लूँगी ही नहीं जी, है ना, मैं वैसा नहीं करना चाहती कि नहीं भी मैं करूँगी, थोड़ी मेहनत और कर लूँगी, थोड़ी मेहनत और कर लूँगी, फिर करूँगी, ऐसे अच्छा नहीं लगता ना कि हम एक प्रोफेशन में हैं। अब जब हम सर मैम कंटिन्यूसली काम कर रहे हैं, बीच में आपका ही फ्रेंड आके आपको मैसेज कर रहा है ग्रुप में, कल भी उन्होंने किया था एमबीबीटी में कि योर टीचर इज टेकिंग माय लेक्चर, दिस इज मैंने रिव्यू पेपर एडिट आपको बताए, है ना, और हम्म, नहीं नहीं मैम, पर नहीं नहीं, मैं उनकी मेंटल लेवल समझ गई मैम, वो मेरे फ्रेंड है, अच्छा मैं उनकी भी रिस्पेक्ट करती हूँ, कोई बात नहीं, उन्होंने मुझे कह दिया, मैं गुस्सा नहीं कर रही। हाँ, बिल्कुल फ्रस्ट्रेट होता है। हाँ, और चलो ठीक है, कोई बात नहीं, मैंने उस चीज़ को लेट गो कर दिया। अब आज अभी भी मैं जैसे 9:15 लेक्चर अटेंड कर रही थी, तो वहाँ पे मैम सर के मैसेज आते, गुड वेरी गुड मैम, है ना, यस मैम, स्माइल बड़ी बड़ी, और फिर उन्होंने कहा स्लीप गुड मैम। तो वो मुझे क्या दिखाना चाहते हैं कि मैंने उनके लेक्चर ले लिए कि तुम सो जाओगी, मेरी नींद होगी, कुछ भी लगा लो। अब मैम यहाँ पे बच्चे हैं। वो है, नहीं नहीं मैम, मैं ऐसा कभी नहीं, अगर मैं मान लो वत्सल सर से नाराज़ हूँ या मैं आपसे नाराज़ हूँ, मैं कभी भी आपको आपके प्रोफेशन में थोड़ी जाके कुछ गलत बोलूँगी। जो नाराज़गी है वो पर्सनल लेवल पे। तो मैंने इस वत्सल सर को यही कहा है कि मैं एक्चुअली मैम, मैं मेरे देखो, मेरे पास 3 है, मैं 2 के लिए प्रिपेयर थी, चलो तीसरा मैंने ले लिया क्योंकि साक्षी चलेगी। अब मैंने ये सोचा है कि मैं 2 तो पूरे करूँगी, जो एमबीबीटी है, क्योंकि देखो अभी 9:15 हो गया मैम, मैं 4 बजे से बैठी हूँ, मुझे भी रेस्ट चाहिए कि मैं एक पहले मैं पढ़ा भी रही हूँ, फिर ऐसा भी स्ट्रेस मेरे माइंड पे रहेगा, मेरे से वो बच्चों से फिर मैम वो ऑनेस्टी नहीं रहेगी। तो मैंने सर को भी यही कहा है कि मैंने देखो 10 दिन के लेक्चर मैंने ले लिए, 10 आवर्स आज मेरे पूरे हो जाएंगे, जो रिमेनिंग 10 आवर्स है ना, वो आप प्लीज़ नंदी सर को दे दीजिए, ये मेरी रिक्वेस्ट है मैम एडी फैब्रिका टीम से। नहीं सर, असल से आप मेरी मैंटेलिटी समझिए, मैं बहुत 3-4 दिन से मैं आपको ये बात नहीं कह रही थी, मैं कह रही हूँ तो मैं कुछ सोच के कह रही हूँ। अदरवाइज को किसी को हाथ में आई मनी किसको नहीं चंगी अच्छी लगती ना, हाथ में आया पैसा सबको अच्छा लगता है। पर मैं खुद कह रही हूँ कि आप मुझे हाफ सैलरी दे देना, हाफ आप सर को दे दो, क्योंकि मैं नहीं चाहती कोई अगला मुझे कहे कि तूने मेरे, मतलब कि मैं सर इस चीज़ में नहीं रहूँगी। आप 10 दिन उसको हम्म, नहीं वो उनसे आप पहले नहीं नहीं मैम, वो वो भी मेरे फ्रेंड है, मैं उनको भी जानती हूँ, मैं वत्सल सर को भी देखी हूँ कि ये भी बहुत 2-3 दिन से इतने स्ट्रेस में है। मैं नहीं चाहती मैम। हाँ, वो तो हमें पता है, सब कुछ बिल्कुल पार्शियल, सब कुछ बिल्कुल ही ट्रांसपेरेंट था, कुछ भी पार्शियल नहीं था। पर जब वो समझने वाला सामने वाला सर भी, मैम वत्सल सर भी बहुत स्ट्रेस्ड है 3-4 दिन से, मैं भी बहुत स्ट्रेस्ड हूँ। फिर मैंने ही एंड में सोचा कि कुछ नहीं होता, 1000 ज़्यादा मिल गया, 2000 कम मिल गया, ये तो आना जाना ही है। आप उनको दे दीजिए थोड़े से लेक्चर, वो भी ठीक हो जाएं। वो ही ना मैम, एक्चुअली मैम शिवानी मैम, आप समझिए, देखिए अभी मैं क्लास ले रही हूँ, पर मैंने आपको फाइनली बता दिया कि मैं 10 आवर्स कंप्लीट करूँगी, मैं और नहीं लूँगी, क्योंकि मैं बहुत स्ट्रेस में हूँ। ऐसा फ़ायदा क्या, मैं फ़ोन स्विच ऑफ कर लूँगी, आप उनको दे दीजिए, मैंने नहीं लेना, बस टू मच हो गया है। हाँ जी। सर मैं आपसे बिल्कुल एग्री हूँ, पर मैं ये स्ट्रेस नहीं लूँगी अभी, और नहीं नहीं, आप रिलैक्स कीजिए, मैं शिवानी मैम से बात करूंगी, पर मैंने यही बात करनी है। अच्छा चलिए। हम्म 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 हम्म। यार नहीं, मेरे से नहीं हो पा रहा, मेरे से 3 नहीं हो पाएंगी, आप उनको नहीं। नहीं। अरे वैसे मैंने साक्षी का भी कर लिया था, अनंत सर को भी तो प्रोटोमिक्स दे ही दिया है, आप उनको दे दीजिए प्लीज। बस मैं हाँ बिल्कुल बिल्कुल। हाँ मैम वो ठीक है, पर मेरे से ले ही नहीं जानी, आप उनको दे दीजिए मेरा हाँ मैं क्लास ले लेती हूँ। ठीक है, ठीक, ओके ओके सर, ओके ओके। Hello, hello, hello. Good evening to all of you. I'm really sorry for little bit of timing because I'm not able to share it in the group. So I hope any one of you please share it in the group so that students will join with us and we will able to start the lecture as soon as possible. Okay. So my voice is audible to all of you. Please let me know my voice is audible. Thank you, Mohammed, and thank you, Simran, for just sending it in a group because I'm really not able to send it in the group. So yes, I welcome all of you in this lecture series. Hope you are all fine and doing great in your life. And my voice is audible to all of you. Please let me know my voice is audible to all of you. Please confirm only once so that we will have no disturbance. Then I have to discuss one two important points just before starting the session of today. So I just want students to join so that we will start the discussion which I want to discuss. Okay. Okay. Yeah. Good evening. Nikita, Priya, Jaguriti, Ritu, Parna, Preetam, Muskan, Simran, Shri Lakshmi, Himanshu, Ankita, Pooja, Roma, Ranit, and we have Mohammed. So I welcome you all in this lecture series. Hope you all are fine. And Shruti, yes, good evening to you also. Hope you all are fine and doing great in your life. Now I have to discuss two, three important points. So let me discuss it. Then I will start the lecture series. The very important point which is there. I know some of you because right now 200 students are there in this MBBT batch. Okay. So right now, as you know, we have 200 students in this MBBT batch, more than 200. But but let me complete my talk first, Himanshu. Then I will let you know. Okay. Uh, so yeah. So as you all know, we have approximately 200 plus students or participants in this MBBT lecture series, but only few of you are able to attend the session. Maybe of your exams, maybe of some another issues. No worries. I am not forcing any one of you to be here continuously or to be here on time. I know these are the online classes, so you are able to watch it at any time when you will be free. Getting my point? So the link is always there in the group. So when you will be free, even at the night, even in the early morning, you have to click on the link and just you have to watch the video. So there is no compulsion regarding the presence of all of you in this lecture. Okay, so like I just post today that I will be sending the link around 9 I just sent. So some of is asking, ma'am, I will be free only by 9:15. It's okay if you will be free by 9:15, no worries at all. But you have to just be there on the link when you will be free. So don't ask me to, ma'am, please be on 9:15. Please, when you will be free, you can watch it at any time, because I have my very busy schedule. I am just continuously sitting in front of a laptop from 4 p.m. So my schedule is very tight. So that is why you have not to asking me, ma'am, please take on that time. Don't worry. You will be, when you will be free, you can have to click on the link and you have to start it. Otherwise, it's okay when I will share the link. If you are free, join. If you are not free, watch it later. The very first thing which I want to clear all of you. Now number 2, as you all not are present on time, or maybe I am not also, so the attendance is not compulsory here, but the homework, the assignments which I gave you, the homework which I gave you have to be done by every one of you, because I am just recording, I am just keeping the record for your assignments. I am just checking your assignments whether you send it to me or not, and according to it you will be provided the certificate. So attendance is not mandatory, but yes, assignment should be submitted within 24 hours. Getting me? Attendance is not compulsory right now here in this live. It's okay if you will want to do it later. No worries, no problem. But assignment is mandatory for all of you. As even I watch in the today's telegram, I'm just watching how many of you have replied, or how many of you have submitted the assignments. The number is very less. If we have 250 students, the assignments are very less. So I request all of you, I am not taking your attendance right here, but yes, I am just going through with your assignments. I am just keeping the record of your assignment. If you are not able, if you will not be able to send assignment on time, then it will create problem to you later on during the certificate. So please, assignment is mandatory, assignment is compulsory. So you have to, like, some students are very good, they are just keeping the assignment on time. I am taking everyone, yeah, you have to submit it in a group. I am taking the record of every of these. I am just going through the group. When I get free, I just check it and I just tick on your name that you submitted it. So it's my request to all of you, and I am the only one. I just asking you, or I am just letting to know you regarding this thing only once. So be ready with it. If you are not able to join this class, if you are not able to be here, no issues, but your assignment should be on time. Getting my point? Your assignment should be on time. Now third thing which I want to discuss, as I came to know many of you have your exams from 16th of December. So how many of you have the exams from 16th of December, or maybe 17th, or maybe 12th of December? Please don't worry if you don't want to take internship, no issues. But even for the journal certificate, assignments is compulsory. For your certificate, the assignment, the submission of assignment is compulsory. Now, how many of you have exams right now in the month of December? Please reply me as soon as possible. Even the students from the genetics batch, they are so much good, they are just keeping the assignment on time. So I request all of you to keep your assignments on time. Yes, please, every one of you, uh, how many of you have exams, and from when it will be start? From when it will be start your exam? From 12, from 16? Okay. Because I get so many messages that you have your exam from 16th or 14th of December basically. Divyansh, you are not added yet. Okay. If possible, send me. But I don't know how to add in Telegram. I am also new to all these things right now. How to add in Telegram? Ask any friend of you to add you in the group, or I will ask sir to add you. I will just writing your name, Divyansh. Divyansh. Bye. Ah, yes, I know many of you have your exams. Vani, you are also not in Telegram. But why you are not in Telegram then? Vani, why you are not on the Telegram? Yeah. Good evening, Anvisha. Nancy's exam is in December. Okay, from 14th of December. Okay, so now the main thing which I want to tell, Dhruvin's exams are from 12th December. Okay, Dhruvin, you are not present in the last session. Why? Because of exams. So yes, today is 8th of December and you have your exam from maybe from the next week from Monday or maybe from Tuesday. So now what I think that this is your syllabus, okay? This is your syllabus. Now there are some topics which are quite related, like this is applications of the recombinant DNA technology part 1, part 2. Some topics are quite related if you are able to see here, like microscopic is divided into three parts but they are together. Similarly, application of molecular biology is together. So what I think that instead of spending the extra day, rather we will try to complete the same topics on the same day. I will take your session a little bit longer. So be ready with it that I will take your session a little bit longer. Instead of 1 hour, it may be 1.5 hours, so that we are able to do two topics which are same together. So by this we will be able to complete your topic by 14th or 15th of December. Okay, so by this we will be able to, instead of 20th of December, we will try to complete your topic by 15th or 16th of December, so that all of you will be able to do your exams also. Even you will get more time for your review paper also. So it is my suggestion to all of you, if you are comfortable, shall we take little bit more time instead of 1 hour only. I just, because when we are just doing it with concentration, I just want to do the same topic with full concentration. So maybe the session will be 20 minutes or 30 minutes longer. So are you comfortable in it if we just take the lecture little bit longer, and instead of 20th of December we will try to complete it by 15th of December, because on Saturday and Sunday definitely I will take 2 hour long session. So I will try to complete it by 15th of December. So are you comfortable with it? Please let me know, so that we will be able to complete it. You will have time for your exams also, and you will have time for your exams also. Even you will get more time for review paper also, because otherwise I am just asking you about review paper, you are not able to understand what to do, whether to take the classes, whether to do the assignments, whether to do the review paper, whether to do the your own final exam. So it becomes very hectic for all of you also. So it's my suggestion only, because I came to know regarding it that you have so many of exams in the month of December, all of you. So if you are, if it is possible, if some are feeling uncomfortable, no issue. If you have 1 hour only, don't worry. Just take the lecture for 1 hour, and the remaining hour you will watch it later also. Because I just want that my students will feel free if possible. So yeah, I can't repeat because I'm just continuously speaking from the 20 minutes. You will get it because I'm repeating the same thing again and again. Otherwise, anyone from the your friends, all the participants, all the students are very nice. They will definitely help you and they will definitely let you know what I speak. So I am just taking your concern because I know you all are mature students. You also have some work to do. Otherwise, what happen if I am giving you assignments also. I am giving you review paper also, and then you have your final exams also. Then it becomes extremely difficult for students also to manage the things. Then I came with this thought only, that instead of forcing you to do review paper, do assignments, rather we will focus on the lecture series right now. You have to submit me the assignments, you have to focus on the lecture series, and later after it you will have ample of time for submitting the review paper, the topics and the abstracts also. So is it fine for all of you? Please confirm once, so that we will be able to complete the syllabus faster, and we will able to complete it within this 14th or 15th of December, so that it will be comfortable, I think, for all of you. Please let me know. You all are free to speak. Not free to speak, rather you all are free to write. so that I just came to know, yes, everything is fine. Yeah, yeah, Preetham, don't worry. Submit your assignments tomorrow also. But it's my request to all of you to try to submit the assignments by 24 hours, because I have to evaluate it also. I have to check it also. Then I have to store, keep the record also. So I hope you all understand my situation also. Ayushi, no worries dear. I am always available to all of you. I will share the PPTs with all of you. I know sometimes I am very fast. Don't worry, just go through the PPTs. If you have any doubt, if you are not able to get anything, no issues at all. We will discuss it again. We will do the Google Meet or something like that, so that I will let you know everything if you want to know. Okay. So don't worry, you all are my students, and sometimes it is very difficult to understand the thing by only once, especially if you are new to that thing. So don't worry, I am here along with you, but I'm just thinking about the whole students, the whole MBBT lecture series, because I know you all are intelligent and you are doing everything on time. Even you are sending me the assignments also on time. Some of you, many of you are not, but it's okay. It's a part of life. So that is why I just think that I just want to do those topics which are related. I just want to do those topics together. Similarly, like today also I'm taking the class of genetics also. So instead of 1 hour or 50 hours, I have taken the session for 1 hour or 20 minutes or 25 minutes. So I just want your 20 to 25 minutes. I hope all of you will cooperate with me. And yes, thank you all. So shall we start now? It's we have discussed. So I have discussed three things. Number one, attendance is not mandatory. It's okay if you are not free. It's okay. Number two, assignment is compulsory for all of you to submit me as soon as possible. Number three, you have to be ready for little bit long sessions, especially on the weekends. That is on Saturdays and Sunday. I will take little bit long session. If you are not free at that time, no worries. You can watch it at later also. So I'm not forcing all of you to be there, but yes, I will deliver my lecture little bit longer. So yes. No, no, there will be not an exam for the certificate. You will get the certificate when you will submit me your assignments on time. That is why I told all of you that I am keeping the record. I just go through all of your assignments. You don't think that you just post it in the Telegram and I will not watch it. I am watching it. I am just, so that is why I request yesterday also to write your name on the assignment, because I am just keeping the record of it. Mudita, I share the PPTs right after the lecture, when the topic is over. I just share right after, but sometimes because of my busy schedule, I was not able to share. So I will just ask in the group, ma'am, please share, please share. Then I will definitely share. Sometimes I forget also. So thanks you all for your cooperation right now. So now we have to start the lecture series. We are already very late. Okay. So we have done basic principles of MBBT. We have done cell culture techniques. Okay. Then we have not done this molecular biology. Rather, we have done recombinant DNA technology. When we are working on the weekends, we have take this part. Now, yesterday we have done separation of molecules by electrophoresis. We have also done electrophoresis. Now, these topics we have just start protein yesterday. Now we have to do how to do protein separation and how to do protein purification. So I will now today my focus is on proteins only. After it, tomorrow we will go with another session, either with microscopy or with molecular biology or something like that. So I will try to do protein session today. Okay. So let me share the PPT. So I am not forcing any of you to be here continuously. You can go anywhere, or you can watch it later also when you will be free. I know you all will watch it, because when I gave you assignment, when I gave you homework, then definitely you have to watch the video also. So yeah, today we have to do proteins part. We have to complete it today, and we have 117 slides. We will try to complete it. So yes, we have done this thing yesterday also. What are proteins? The introduction, that how the proteins are formed, the types of protein. We have fibrous protein, we have globular protein, we have membrane protein. So these are fibrous proteins, globular. And what is the homework? What is the homework which I gave you all? I hope you all have done it now. So please, yeah, I gave you the difference between horizontal and the gel electrophoresis, and what is the need of polyacrylamide instead of, when we have agarose, what is the need of polyacrylamide. So only few of you have sent me your assignments. Otherwise, rest still are pending. So some of you have written it very beautifully. Ankita Jaiswal is here, and lots of other students are Lakshmi Krasna. So just and we have John Weasley also. So I will give you little homework today also, so you have to complete it. Okay. So yeah, so this is the fibrous proteins, globular proteins, membrane proteins we have done yesterday. Types of proteins: primary structure, secondary structure, tertiary structure, quaternary structure. I will share the PPT and you will go it by your own self. Yeah. Is it visible? Yeah, it's visible. Okay, okay. Yes, yes. Some of you already sent me. I know. Thank you for your code. And then yesterday we have done extraction. So you have to keep three things in mind. We have extraction. We have extraction, or you can say isolation. Then we have quantification, and then the last is the purification. So this is the protein extraction. This is the protocol which is there. Like if you want to extract the proteins from the plant, you want to extract it from the animals. So we have different protocol. Okay, for animals we have different protocols. If you want to extract it from fungi, we have different protocol. We need different types of materials for it. So just go it by your own self. You have to read it. Now this is the protein extraction from the bacteria. This is the procedure of it. Now yeah. So today, so we have done protein isolation, or you can say protein separation. Now we have another thing which we have to discuss, which is the protein separation, basically. How to separate the proteins, how to do the protein separation. So this is one method of protein separation which is known as SDS phase. It is also known as sodium do-decyl sulfate polyacrylamide gel electrophoresis. This is known as sodium do-decyl sulfate polyacrylamide gel electrophoresis. So it is a common technique used to separate, visualize, and therefore compare the relative amount of individual polypeptide chains contained in different fractions. So basically this SDS phase is basically used for the separation of the protein. Getting my point? For the separation of the proteins, and then we have to visualize it under the UV light or somewhere. So we have to do the separation and we have to visualize it. Therefore, we are able to get differentiate the different types of proteins according to their molecular weight. We are able to separate those proteins. So it yields the mass of each individual submit derived from the denatured complex. So SDS phase is basically for the separation of the proteins. Now what is the procedure? A small sample from each fraction is fixed with an excess of SDS, beta mercaptoethanol and bromophenol blue. So these are the three you can say 3 chemicals which we need, or you can say we have SDS, which is sodium glutathione, we have beta mercaptoethanol, we have bromophenol blue. So each mixture is then placed in a boiling water bath for several minutes. In the end, each polypeptide chain is unfolded into a negatively charged rod shaped complex with a relatively constant charge to mass ratio. So this is denaturation. This is basically denaturation, when we are just boiling the mixture to denature the proteins, or you can say to unfold the proteins, and then at the end we will get negatively charged rod shaped complex with a relatively constant charge to mass ratio. So this is again, this is electrophoresis. Electrophoresis is also for the separation of the proteins and for the separation of the biomolecules, the DNA, the nucleic acid. So this is electrophoresis. This is horizontal electrophoresis. Now take a solid rectangular gel of acrylamide that is cross-linked to a desired mesh or size exclusion limit. A small helicoid for each denatured sample is transferred to separate wells of a gel. So we are using the combs in which we will add the sample we will add in these wells, and then we have to apply the electric field. We already discussed electrophoresis in detail. Yesterday we discussed it, so I will not repeat here. So we are just using the electric field, we are just using it, and in the wells we have the sample and they will migrate. So this is the direction of movement, they will migrate. So an electric field is applied across the loaded gel with positive pole positioned in the opposite sides of the sample. So we have positive, we have cathode, we have anode, and then the sample will move. So electrophoresis we already discussed yesterday. Now this is vertical electrophoresis. Now here what happens, negatively charged complexes get dried through the mesh of polyacrylamide, which effectively filters them according to the length of their stroke radius as they wiggle through the porous matrix. So this is the gel, and if you are able to see, we are able to get the bands also, we are able to get the fragments also. So how the molecules will move? So obviously the smaller fragments, the size, the protein molecule whose size is small, they will move fast as compared to the larger one. So larger one will take the time, but the smaller one will move fast under the presence of a electric field. So small particles, they will move through the gel more rapidly than the larger ones. The electrophoresis is visually followed. Power supply is shut off before any protein in the sample reaches the bottom of the gel. Yes, one thing you have to keep in mind, that there is a limit of everything. There is a limit of electric field which we have to provide. Otherwise, what will happen? These bands will move out of the gel. So we have to keep the timing on for 30 minutes, for 40 minutes we have to run the gel, and then we have to keep the power supply off. Now after it, there is one more thing which is there, which is staining and de-staining, especially in case of SDS. Now what is staining and de-staining? What is electrophoresis right now? Separation of the protein molecule. So after it, the entire gel is soaked in a stain solution. Let me show you the PPT. Yeah. So when we just apply the electric field, after it we have to soak the gel in a stain solution containing a dye that tightly binds to the backbone of each denatured polypeptide chain. So this is the dye which we basically use, which is Comessi Brilliant Blue. So we have to soak the dye in the we have to soak the gel in the stain solution. Excess stain is then washed from the gel by soaking it in a de-stain solution. So first we have to add it in a stain solution, and later we have to add it in a de-stain solution long enough for the blue stain bands of each polypeptide to be visualized against the clear colorless background gel. So why there is a need of staining and de-staining? This is your homework now. Why it's okay, we have done the electrophoresis. No. After electrophoresis we are getting the bands. We are able to get the bands. But then after it we are doing the staining. That means we are giving the gel some color. We are giving some color to the gel. After staining, what is the need of destaining? Getting my point? So even I suggest you to go on YouTube to watch how they are just doing the SDS page or something. Even I will show you the videos also, no worries. So this is your homework. We are doing why there is a need of staining as well as destaining. If staining is done, why there is a need to do destain? So this is the thing. If you are able to see, this is 2 SDS paste gels after a completed run. So page of a sample protein stained with a Comessi blue. So that is why the color is blue. These are the bands basically. The color is blue because of the presence of the dye. So after staining, different species, after staining as well as destaining. So after staining, different species biomolecules appear as a distinct band within the gel. So if you are able to see, this is the gel, this is the molecular marker which we run, and we are getting these bands. If you are able to see, we are getting these bands. So it is common to run molecular weight size. So those which will come fast, they have the low molecular weight, and those which will remain on the upper side, they have the high molecular weight. So it is common to run molecular weight size markers of known molecular weight in a separate lane in the cell to calibrate the gel. So approximate molecular mass of unknown biomolecule is determined by comparing the distance traveled according to the marker. So these are the molecular markers, and we know the size of these bands. We know this is of 100 kilobase pair, this is of 500 kilobase pair. We know the size of these markers. So when the band is present here, we will able to know the size of that particular band. Are you getting me? Let me check. So everything is fine right now? Yeah, you have to answer me, but not right here. Later on you have to answer me. So up to now, is it clear to all of you? Okay. So let me share the PPT. Actually, dear, bromophenol is there in the gel. When we are loading the sample, at that time we are using the bromophenol. Comessi blue is for the staining, when we want to see the band, because the gel is transparent. Getting my point? The gel is transparent. So when the gel is transparent, when we are loading the sample, at that time we need the bromophenol blue. After it, we run the gel for 40 or 30 or 40 minutes. Now the gel is transparent and bands are not completely visualized. So we have to stain it with the Comessi blue. So at that time, when we have to stain it, we need the Comessi blue dye. So that is after it we have to do the staining and destaining. So these on this side we have the molecular markers, and we know the size of each these bands. This is of 100 kilobase pair, 500 kilobase pair, 700, 1100 kilobase pair. So when the band is there in front of it, then we will able to know the size of that particular protein molecule. So approximate molecular mass of unknown biomolecule is determined by comparing the distance traveled relative to the marker. So this is basically what this is the separation of the proteins. Separation of the proteins by SDS, separation of the proteins by this electrophoresis. After separation of the protein, what is the next step? After it, we have to check the concentration of the protein by spectrophotometer. Spectrophotometer, all of you must know about it. This is the basically this is used for the to know the concentration of protein. That means how much pure the protein sample is, or how much contamination is there in the sample. So this method is recommended for the pure protein solutions. So we want to know whether our protein solution is pure, or is there any contamination also, like some contaminations also, or the sample which we have is the whole protein molecule. So protein in solution observes UV light with absorption maximum of 280 and 200 nanometer. Amino acids with the aromatic rings are the primary reason for the absorption peak at 280 nanometer. Now this is the absorption maxima at 280 nanometer and 200 nanometer. So at that range we will able to absorb the protein. Amino acids with aromatic rings at the primary region for the absorption peak at 280 nanometer. So peptide bonds are primarily responsible for the peak of 200 nanometer. You have to keep only one thing in mind, that when we will get the range, when we will get the protein, the results in this range from 200 to 280 nanometer, that means we have the protein sample. So this is a spectrophotometer. This is a cuvette in which we have to put our sample. Then we have to put it here in the machine and we will get the reading here in this graph. So this is a procedure. Warm the UV lamp, adjust the wavelength to 280 nanometer, then calibrate to zero absorption with buffer solution only. So first we have to calibrate it with buffer solution only. Before adding the sample, we have to add the buffer only to make the whole thing neutral. So measure absorption of the protein solution, adjust the wavelength to 260 nanometer, calibrate to zero absorption with buffer solution only. Mere absorption of the protein solution. Then after first of all we have to calibrate the machine by using the buffer sample, and then later we have to measure the absorption of the protein. So this is the concentration, how we are able to get the concentration and the results. So pure protein of known absorption coefficient for 1 centimeter path length. This is the mathematical basically visualization, how to do, how to concentrate, how to know the concentration of the proteins and everything, and how to know the contamination if there is any contamination, how to calculate that particular contamination also. So this is only the calculation part here. After quantification, now this is when we are able to quantify the proteins, when we are able to know the concentration of the protein. So first we have done separation of the proteins, we just separate the band, we are able to do the separation only. After it, now we have done protein concentration or protein quantification. Now after it we have to do protein purification. How to purify the protein. Suppose I just want to work on specific molecular weight protein. So how to purify that particular protein, how to purify. Now why do we need to purify the proteins? Because sometimes we want to work on a specific protein, we want to discuss the structure and function of the specific protein. For that particular we need to isolate, we need to purify that particular protein only. Okay, after it, to study protein regulation and protein-protein interaction. So we have to study the protein regulations and the protein-protein interaction which are there. So if you are able to know for the proteins only, to study the proteins only, the special field of science is there which is known as proteomics, in which we will focus on just on proteins and proteins only. So for those we just need to purify the protein. To produce antibodies, proteins are used in assays also. They perform structural analysis by X-ray crystallography. Now before purify the proteins, these are some sources from where we will get the proteins. From the whole organism, from the organs, from the embryos, tissue culture, even proteins from the expression system also. So we will able to get the protein from these sources. Now this is again the protein isolation step. I am not discussing protein purification here, I will discuss it, just wait. This is again the protein isolation part, the very first part which I discussed yesterday. For protein isolation, there are so many things. We have chromatography, ultrasound homogenization, French press, cryogenic grinding, lysis buffer, which we have discussed yesterday also. These are the steps which are needed in the isolation. We need homogenization, centrifugation, filtration, and at the end we will get the protein molecule. Okay, this is the homogenization, the equipment which is there in the lab for the separation of the protein. This is the centrifugation. You all know the machine in which we have to put the sample, and then we have to rotate it very fast for 15 to 20 minutes, and at the end we are getting the pellet. So we will have to discard the supernatant and we have to take the pellet only. Then this is another technique which is French press. This is also for the isolation of the proteins. Cryogenic grinding is basically what? When we are using liquid nitrogen, the temperature is minus 196 there, and we are using this for the separation or for the homogenization of the protein. These are lysine buffer which are used. Then stabilization of protein. Yes, one more thing is which is quite important. When we want to isolate the proteins, when we want to extract the protein, there is some care which must be taken to preserve the protein structure and function after it is removed from its natural environment. So if you want to take the protein structure and function, you must have to take everything in mind: the temperature, the pH, there will be enzymes also. So what is the pH? Basically, to prevent denaturation or loss of functions, proteins are placed in a buffered solution. So we are just keeping them at the pH which is their native pH. So we are just dealing with the buffer solution. Temperature, always take the temperature in minus degree Celsius. Just like yesterday also, we put the sample in the ice bucket so that the temperature will not raise. So we have to keep the temperature as low as possible, even in the presence of liquid nitrogen. In the liquid nitrogen, the temperature is minus 196 degrees Celsius. Even we need some, we have to inhibit some proteases. These are the enzymes which will break our protein, and we don't want to kill the protein. So we have to keep these things in mind. So these are some isolation of protein. These are some methods of isolation. We have salting out, we have dialysis, we have chromatography, electrophoresis we have done, we have isoelectric focusing which we have to discuss in detail, we have chromatography. These all things are basically for what purpose? For the isolation of the proteins. So this is again the separation of the proteins. If you are able to see here, this is dialysis. Now what happen in dialysis? Basically what will happen in the dialysis? If you are able to see in this diagram, we are just running the samples in this tube. Now there is a semi-permeable membrane in between. So there is some sieve size. There is some sieve size. So only specific type of molecules which are smaller than this sieve size, they will be able to pass out. If you are able to see here, only we have red and we have blue particles, but only red particles whose size is small, they will be able to move out. The blue one will remain inside the same place. So this is how we are able to isolate, or we are able to separate the proteins. Again, if you are able to see here, this is centrifugation. Before centrifugation, the machine in which we are doing it for a very long time at a very high speed. So if you are able to see here, this is a mixture of proteins. Everything is not separated right here. But after centrifugation, what we will get? The heavy particles will settle down, and the lighter one, they will remain on the upper side. So this is again the method of separation of the proteins. Then we have another term which is chromatography. I hope all of you must be aware of chromatography. If you have must done in your theory classes also. You must have done it in your theory classes also, so I will not take it much longer. Basically, chromatography is a technique for separating the mixtures into their components in order to analyze, identify, purify, quantify the mixture or the component. So in chromatography, we are able to separate the mixture. This is a mixture. We have yellow particles, blue particles, red particles. With the help of chromatography, we are able to separate them all. So according to their molecular weight, they will be separated. We have different types of chromatography. We have liquid chromatography, we have gas chromatography, paper chromatography we have done yesterday also. Thin layer chromatography is there. I will not do these chromatographies in detail. I will share the PPT. You will read it by your own self. Then we have column chromatography. The only thing which you have to keep in mind, that chromatography is there for the separation of the protein molecules according to their size. You have to keep this thing in mind only, that's all. Okay, so like if you are able to see, these are the porous polymer beads, these are the gel beads. We are just putting our sample in it, and slowly with the passage of time, we are able to isolate the protein molecules by their size, Larger particles will take more time and smaller particles will run fast. Similarly, as we have done in electrophoresis, this thing is same again. Now this is gel filtration chromatography, in which we are using the gel beads. So if you are able to see in this black and white diagram, the smaller particles, they will move fast and the other one will move slowly and slowly. Okay. The salting out is again another method for the separation of the proteins. Dialysis is here according to their porous size. If you are able to see this diagram also, so there is a specific porous size of this blue, this permeable membrane, semi-permeable membrane. Only the small ones, which are these orange one, they will be able to pass out. The green one will remain inside. So this is dialysis. Gel filtration is that when we are able to separate according to the size. Now what happens in this gel filtration chromatography, this is also known as molecular exclusion chromatography. Here we have two things. If you are able to see, the blue one, the smaller one, they just get embedded in the gel pores. They just get embedded in the gel pores, and only red one, which are quite large in size, they will move out. So it's not only that small one will move out first. No, there is no any type of criteria like this, because these gel beads, they are porous in nature, so they will just absorb the smaller ones and the larger one will move out. So if you are able to see, these small molecules, they will add in these resin beads, and the larger one, which is basically the proteins, they will move fast. So the beads are made up of insoluble but highly hydrated polymers such as dextran or agarose or polyacrylamide. So these are the polyacrylamide beads basically. Sample is applied at the top of the column consisting of porous beads in the buffer. Small molecules of the sample can enter these beads, but larger ones cannot. Small molecules are distributed inside the beads. So small molecules, they will be absorbed inside the beads, and the larger one will be allowed to move out. So the larger molecules flow more rapidly through this column, while the smaller molecules take a longer path to move out. So is it clear to all of you up to now? Let me check it. Everything is fine. I hope you are not feeling bored and you are just feeling good enough right now. Everything is fine till now. Please let me know. Yeah, please answer your friend's query. If I am not able to answer in between, please, if you have some knowledge, knowledge should always be shared. If you have, you are free to share your knowledge also. Yes, please let me know. Is it are you comfortable till now? Everything is clear to all of you? Please let me know. So is it clear to all of you? Are you feeling comfortable? Shall we continue, please? If possible, shall we continue a little bit more? All of you, please reply. I'm just doing it for your own self, for yourself basically, so that you will be free a little bit early in the remaining days. Up to now, are you comfortable? You all should reply, please. Yeah. Thank you all. Thank you, Afreen, Nilima, Roma, Ria, Muskan, Romit. It will be, I think it will be nice if we will interacting personally and I am able to see all of you, because by your names I am not able to get your face. I am not able to know how you are expressing right now. Hope that we will meet on. I will be able to see all of you. Maybe on the last day we will do Google Meet or something, and then we will have to keep your video on also. So we have Harshni, Saurav, Ahmad, Mohammed, Kusturi, Shreya, Soumya, Vani, Microbial News, Soumya Deep, Monojit. So hope we will definitely, I will definitely want to see all of you. So I just want that we will, on the last day, we will try to interact with each other on Google Meet. so that we will able to see, I will able to see all of you. Okay. So all of you, are you getting it properly? Everything is fine? Yes, we have Srishti, we have Ikta, Shreya, Jagruti, Rubin. Okay, okay. Okay, good, good, Nancy, Nivedita, good, good. Okay. Shreya, Pritam, Divyanshi. Okay. So this is basically chromatography is basically for the separation of the protein molecules, either the large one or the small one, but we have to separate it. Okay, but yes, chromatography takes lots of time because this process is very slow. If you are able to see here, for this, when we have to put it here in these tubes, this step is very long. It's not a simple step. This step is a very long step. So this is our gel filtration chromatography. This chromatography is quite important, and this is used these days also. So gel filtration chromatography is quite common and important also. The another chromatography which is very important, which is known as ion exchange chromatography. So here the separation of protein, like in the gel, we have the resin beads which will absorb some small molecules, but here in ion exchange chromatography, the proteins will be separated on the surface through ionic charge. So proteins with very similar charges can be separated. Ion exchange columns utilize the principle of ion exchange chromatography. So this is the ion exchange chromatography principle. Here the separation is basically on the basis of ions. Here, the separation of molecule is on the basis of ions. So, positively charged protein binds with the negatively charged beads. I will show you here. Yeah. So, here we have a column in which we have the beads attached. We have the column on the surface of which we have the suppose negatively charged or the positively charged. The beads are attached here, like we have resin beads. Similarly, we have the beads which are attached here. So positively charged proteins, now when the protein molecules will move, the positively charged protein, they will bind with the negative one, and the negatively charged proteins, they will bind with the positive one beads during the flow. So if you are able to see, this is the chromatographic tube. On the sides, on the edges, we have the beads present, and on these beads we have certain charges. Either they will be of positive charge or they will be of the negative charge. So ion exchange chromatography relies on the interaction between oppositely charged ion exchangers and the protein sample. So if the bead, if you are able to see, if the bead is negatively charged, then the positively charged protein will interact here. So we have two types of exchanger. Basically, we have cationic exchanger, we have anionic exchanger. So if in the tube, in the beads, in the tube we have the negatively charged beads are present, then it is known as cationic exchanger. Suppose here the beads are positively charged, then it is known as anionic exchanger. So we have in an ionic charge, we have two types of tubes. One is cationic exchanger, in which we have carboxymethyl group attached, and another is an anionic exchanger, in which we have the diethyl aminoethyl group attached. Let me explain it a little bit more here. So ion exchange reagents contain the charged groups. If these are acidic in nature, they interact with the positively charged proteins and they are called cationic exchangers. So cationic exchangers, they will interact with the positively charged proteins. If you are able to see here, this is the cationic exchanger. The negative charge it carries. So the cationic exchanger will carry the negative charge and it will interact only with the positively charged protein. If we have a anionic exchanger, If these groups are basic in nature, they interact with negatively charged molecules. So if we have a anionic exchanger which carries a positive charge, this is known as anion exchanger. It will bind with the negatively charged protein. So we in the sample we are adding positively charged proteins. Also we are adding the mixture of protein. So if the beads are negatively charged, they will attract the positive proteins. If the beads are positively charged, they will attract the negative protein. Let me show you the diagram. So, in the ion exchange, the interaction is based on the net charge and is less specific. The reasons is either negatively charged or positively charged. So, if buffer equilibration exchange reagent is found to counter ions, a cationic exchanger is usually bound to sodium or potassium ions, and an anionic exchanger is usually bound to the chlorine ions. So, we have two types of exchangers. We have cationic as well as in the cationic we have carboxymethyl cellulose. In case of anionic, when we have positive charge, we have diethyl amino ethyl DEA. So these are the four steps which are there in the ion exchange chromatography. This is your homework also. Please, any one of you, please write the homework because I forget at the end. You have to write it anywhere. So you have to tell me about ion exchange chromatography. You have to tell me the four important steps which are there in ion exchange, and even you have to draw the diagram of ion exchange chromatography. So yes, these are the four steps of ion exchange chromatography. We have equilibration, we have sample application, we have elution, we have degeneration. Now what is equilibration? It is a stabilization of ion exchanges with oppositely charged ions. Let me show you. So it is the stabilization of ion exchangers. Suppose the ion exchanger is cationic. Then if it is positively charged, it will be stabilized by oppositely charged ion, either by chlorine or either by sodium. After it, when the sample the exchanger gets equilibrated, after it then we have to add the sample. So sample application and wash, protein bound to the ion exchangers remain attached while other gets removed during the wash. So if we have cationic exchanger, if we have positive on the sides, negative proteins will bind only. So suppose this is the exchanger which we have the cationic exchanger which carries the negative charge. So what will happen now? If it is negatively charged, only positively charged protein will bind to it, and the negatively charged proteins will move out from the exchanger. So this cationic exchanger, it will hold only the positively charged protein. Getting my point? So this cationic exchanger, it will hold only the positively charged protein. So first is equilibration, number two is sample application. When we are adding the sample, then after it we have to wash it so that the negatively charged proteins, they will wash out, they will move out. Now elution, removal of bound protein from the ion exchanger with the help of increased concentration of elution buffer. After it we have to add the elution buffer, and the fourth step is degeneration, preparing the ion exchanger for the next round. Let me show you now. So this is the very first step. We are just we have done the equilibration, and after it we are adding the molecules. The negative charge will wash out, the positive charge will bind. So ion exchange chromatography, to elute our protein of interest, add buffer of now if you are able to see here, now this is the third step. This is the third step in which we have to do the elution. We have to add the buffer. So now these red one, they will move out. Now this is our protein which is our point of interest. So what we have to do now? To elute, now we just want to remove this bonding so that we will separate our protein of interest. So to elute our protein of interest, add buffer of higher salt concentration. So it is negatively charged. This CM cellulose cation exchanger is negatively charged. Now what we will do? We will add sodium, which are of high concentration, high salt concentration. So sodium will interact with the cation resin, and chlorine will interact with the positively charged. So sodium will interact here and chlorine will interact here. What will happen now? They will form this complex, and our desired protein will be move out. So we just want our desired protein. So this is our third step. To elute our protein of interest, add buffer of higher salt concentration. Sodium will interact with the cation resin and chlorine will interact with positively charged protein to elute out of the column. So I hope you get it. So first of all, we just want that our protein interact with the cation exchanger. When it interacts, it's okay. The undesired proteins will be washed out. After it, when we want our desired protein, we will have to add our buffers also, so that we will able to isolate the protein of interest. So this is ion exchange chromatography. I hope you get it. You have to completely write it, everything, the four steps: equilibration, sample application, elution, and what is regeneration, when we are just preparing it for the next round. So proteins will bind to the ion exchanges with different affinities. As the column is washed with a buffer, proteins will have already relatively low affinities for the ion exchange region, will move faster to the column. The greater the binding affinity of a protein for the exchange column, the more will be stored in the eluting of the column. Proteins can be eluted by changing the elution buffer with higher concentration. So same thing which I already told you, this is the same thing. So this is the diagrammatic representation. If you are able to see how, when first of all we are just adding low salt elution buffer, we are just preparing the chromatographic chamber, we are just preparing the exchanger. After it, we are just adding these are some steps, like we are adding low salt and we are adding high salt. So these are the steps which are there in the ion exchange chromatography. Now these are some of the exchanges which we are using in our routine use. We are using DEA cellulose, we are using CM cellulose, which is known as cationic exchanger. This is an ionic exchanger. Okay, so they have some ionizable group which are attached along with it. So you have to know what are these basically. So this is an ionic exchanger. If you are able to see, again you are able to see, we are able to separate the protein molecules by electrophoresis. Gel electrophoresis we have already discussed yesterday. I will not do it right now. Now we have page. Yeah, page is little bit totally different from today's class. Page is, I want to give little bit more time on page. I will not discuss page right here. It needs a little bit more time. This is again ionic exchange chromatography. This is affinity chromatography. So here there is some, in this basically we have some, like we have enzyme substrate reaction. Similarly here they will bind with the desired molecule, and later on they we will able to elute the undesired molecules. But when we want our desired molecules, so these pink one are our desired one, they will be eluted later by using the high salt concentration. So affinity is basically like we have enzyme substrate reaction, same we have this affinity chromatography. Also known as binding affinity. So if you are able to see, you can see the structure of the beads, and only the respective molecules will add in these structures. So if you are able to see, the target will add in these structures and unwanted will be moved out. So later on, so first step is loading, then separation, and later is elution. Now when unwanted will move out, now only the desired molecule, by adding the high salt we are able to extract the desired molecules later on. So affinity molecule like enzyme substrate reaction, antibody antigen reaction. This is affinity when we have the reaction for a particular thing. Suppose if you are able to see here the round one, so only blue one able to bind here. The enzyme substrate reaction, you all know, I know. So this is enzyme substrate reaction. This is affinity chromatography. This is again a mean of affinity chromatography when we are using antibodies and antigen antibody reaction basically. This is gel filtration, we have already discussed before also known as size exclusion chromatography. So according to the pore size we are able to, yeah, according to the pore size we are able to remove those things. So is it clear to all of you? Please let me know, please, are you getting it or not? Thank you, Somyadi. Thank you for your appreciation. It means a lot to me also, because I am also doing lots of hard work for all of you, and I want to keep all these things simple so that you will able to get it and not feel bored, because I know some of you are totally new to it. So I just want to keep it very simple, and it will be an honor if you all are getting it, and it will be so nice, and it is so nice from all of you. Thank you all. Definitely, Yogita, no worries. You have to go it by your own self. If you will not able to get it, then I will definitely help. Don't worry. Because right now you are not in front of me, so it's little bit difficult for me also to keep my pace on the same pace. And even the time is getting fast now. This is 10:30, so that is no. It's a great honor that you are still here along with me for such a long time. And I just want to thank all of you to be here along with me. I think today is our longest session also. We are here around from maybe from 9 or 9:10 p.m. So yes, now please any one of you please revise the syllabus, not the syllabus, the homework, because you have to these chromatographic techniques. These are very important. So you have to tell me about ion exchange chromatography number one. Then you have to tell me about affinity chromatography, and the last is the gel filtration chromatography. So you have to tell me the three very important chromatographies in your syllabus. Even in your future also they will help you. We have ion exchange chromatography, we have affinity chromatography, and we have not gel electrophoresis basically. We have affinity chromatography, and the last is the gel filtration chromatography. So you have to, yes, gel filtration chromatography. So these three chromatographies are there in your homework. I know you all will do it definitely. Answer, no, there is not any specific limit of your answer, but yes, the answer should be like that. You should be comfortable. The answer is only that one, that how much you learn basically, how much you get basically. The answer should be like that you are the teacher and you are just giving it to your student. Okay, you are not giving it to the teacher, rather you are the teacher and you are presenting it in front of someone. The answer should be like that. Okay, I don't know whether you get it or not, but this is the thing. So yeah, so this is any one of you please write the question, so that and please anyone please type it in the Telegram also. And I will some students are there definitely who are helping me, and I will check all these things by this lecture series, and I will definitely ask the team to provide the student who will help me for to give him or her the appreciation award from my side also. So please help me, in these things also, in maintaining the questions, in answering the queries. Some students they will ask the same question again and again, and I'm not able to answer. I'm really sorry. So I just want any organizing secretary, anyone from you, who will definitely get the special appreciation award from my side, from the team, who will help me. So I just want one organizing secretary. If anyone, he or she is willing to become the organizing secretary, it will be so sweet of you, and definitely he will get some special award also. Sapnil, I am unable to repeat because I forget. I am just continuously speaking from the last 1.5 hours. So please, I think anyone will, the organizing secretary will let you know. But now I am totally exhausted. So please, any one of you, please write the questions. Please, I also not know what are the questions are. What is the first question which I tell all of you to complete? Please write the questions. Any one of you, if possible, please write it. Yeah, Muskan, this is the last question. Ion exchange, gel filtration, and affinity chromatography. Very good. What are the another questions? Please let me know. The questions which I asked you in the very beginning. What is the question which we have to Yes. Thank you, Suchita. The very first question is, if there is staining, why there is a need of de-staining? Yes. Thank you, Shafaq, Shreya. Yeah. What's the need of staining and de-staining? Yes. And what is the second question then? Then the second question is the same. Yes. So this is your assignment. You have to, yeah. So you have to tell me about SDS page. That means the need of. So you have to tell me three things basically. The SDS page. That means the need of staining and de-staining, and the complete SDS page which is there, and the three chromatography which is there. So only two questions are there. SDS page, what is the need of staining and de-staining, this is together, so you have to tell me about SDS page also, and another is the three chromatographic techniques only. So that is all from my side for today. I think you have lots of homework to do also. So yes, best wishes from my side. Take care of yourself. Yeah, staining and de-staining, and along with it you have to tell me about SDS. So my first question is basically what is SDS, what are the components of SDS, SDS complete, and the need of staining and de-staining, and number two is the gel filtration. So you have to tell me about the gel filtration also, the gel filtration techniques which are there. Hello? आ जाओ तुझे आ जाओ तुम्हें मान लो. So I think that's all for today. It's more than sufficient. Take care of yourself. Good night. Good night. Take care all. Please be safe. Be happy, all of you. Take care, all of you. Bye bye. Good night. It's already very late. Today is 10:30 p.m. So okay. Best wishes from my side. Bye bye. Yes, microbial news. Thank you so much. Thank you so much. I forget the second step, second question, which is the four steps of iron chromatography. No one just remind me. So you have three questions. Microbial news. Thank you a lot for your help. And just post these three questions in the group also. So it's not only staining and destaining. Rather, we have the SDS completely. Please, thank you, Microbial News, thank you, Harshni, Nikita, Romit, Shruti, all of you, thank you for being here along with me. It means a lot. Take care. Okay, bye bye. Okay, thank you all, thank you. Good night all. Bye bye, take care.